Welcome back to Acing AP Biology. In this lesson, we will be discussing the cell cycle. The cell cycle can be divided into two stages: interphase, which takes up 90% of the cell cycle, and the mitotic phase, which takes up the other 10%. Interphase can be divided into three phases: G1, which is gap 1 or growth 1, S phase, and G2, which is gap 2 or growth 2. Interphase also consists of several checkpoints to ensure that the cell cycle is proceeding properly. In interphase, DNA exists in the form of chromatin. The mitotic phase can be divided into mitosis and cytokinesis. Mitosis has several stages: prophase, prometaphase, metaphase, anaphase, and telophase. Cytokinesis involves the division of the cytoplasm. In G1, the cells are the smallest because they are coming from the mitotic phase. Cells grow in size in G1, but the size of the nucleus and chromosomes remain the same. Keep in mind that cells cannot grow too large because they need to maintain a large surface area to volume ratio. The protein cyclin is also produced in this stage and has an important role in G2. Then comes the first checkpoint, also known as the restriction checkpoint. It is the most important checkpoint in the cycle. Once a certain surface area to volume ratio is reached, the cell passes the restriction checkpoint and it becomes committed to cell division. This ensures that cells do not grow too large. If the cell does not pass this checkpoint, it enters the G0 phase. This phase is for non-dividing cells, like mature muscle cells and nerve cells, which seldom divide and cannot re-enter the cell cycle. After G1 comes the S phase, where chromosomes are copied and DNA is replicated. Next comes G2. G2 involves the preparation for mitosis, increasing the number of organelles and cytosol. The second checkpoint is known as the G2 checkpoint. The G2 checkpoint involves MPF, which is known as the maturation or mitosis promotion factor. MPFs are composed of cyclin and cyclin-dependent kinase, or CDKs. CDKs are always present, and cyclin is not. Cyclin binds to cyclin-dependent kinases to form MPF. When the cell builds a certain number of MPFs, the cell passes the G2 checkpoint and can enter mitosis. Mitosis is the division of the nucleus into two identical nuclei. After mitosis, cytokinesis occurs, where the two new daughter cells form due to the division of the cytoplasm. The third checkpoint occurs in the mitotic phase and is called the metaphase checkpoint. This occurs when the centromeres of the chromosomes line up on the metaphase plate. Each centromere has to be aligned on the metaphase plate in order for mitosis to continue. Once the checkpoint is passed, MPF sends a signal to trigger the production of separase, an enzyme that separates cyclin and CDK. At this point, the sister chromatids separate and anaphase begins. Cyclin is destroyed. After the completion of the mitotic phase, the cycle begins again. In order to maintain proper growth and development, the timing and speed of mitosis is crucial, and so there are various mechanisms that regulate the cell cycle. The rate of cell division varies with different types of cells. For instance, skin cells divide very often, whereas mature muscle cells rarely divide. The cell cycle control system consists of checkpoints to make sure that the cell cycle is occurring appropriately and preventing mistakes. As I mentioned earlier, the three checkpoints include G1, G2, and the metaphase checkpoint. MPF acts as a mitotic clock, regulating the changes in the dividing cell. It is composed of cyclin and cyclin-dependent kinase. It mediates the progression of G2 and also is involved in the advancement of mitosis during the metaphase checkpoint. There are also many internal and external signals that regulate the cell cycle. An internal signal includes the signaling during the metaphase checkpoint, where the centromeres must be lined up on the metaphase plate for the next stage of mitosis to continue. Kinetochore protein in centromeres are involved with signaling in this checkpoint, ensuring that both daughter cells receive equal number of chromosomes. External signals include both chemical and physical factors. Chemical factors are critical because they prevent cell division from taking place when essential nutrients are not available. For instance, growth factors regulate when cells should divide, and it is a highly controlled process. There are two types of physical factors: density-dependent inhibition and anchorage dependence. Density-dependent inhibition is the phenomenon where cultured cells will stop dividing once a single layer has formed. Any vacancies in that layer will be filled immediately. This prevents too much cell growth. Cancer cells do not follow density-dependent inhibition and divide continuously. According to anchorage dependence, cell must adhere to what is called the substratum, which is either a culture dish or the extracellular matrix, in order to divide. Cancer cells form tumors that interfere with the functions of the body. A benign tumor involves abnormal cell growth that remains at the original site and can be removed with surgery. A malignant tumor moves to other parts of the body and becomes invasive. When tumors become malignant, it leads to cancer. Cancer cells do not follow control mechanisms or regulatory signaling. Cancer cells divide indefinitely if nutrients are present. They also do not follow density-dependent inhibition. They stop dividing at random parts of the cycle rather than obeying the checkpoints. That's it for this video. In the next lesson, we will go through the stages of mitosis. See you then. For more bio content, hit subscribe.